Transcription. Next topic, that is the transcription. Transcription and translation, these are the two steps in protein synthesis. Transcription and translation, these two steps are involved in protein synthesis, and these two are involved in central dogma of molecular biology. According to Crick's central dogma of molecular biology, in that two steps include from DNA to the mRNA, that is called transcription. From mRNA to protein formation, that is the translation. So, first step in this protein synthesis or central dogma of molecular biology, that is transcription. What is transcription? DNA dictates which type of protein to be synthesized. That information taken by RNA from the DNA and prepare that synthesize that particular protein. Formation of mRNA by receiving genetic information from DNA is called transcription. The procedure of obtaining genetic information from DNA by mRNA, that is called transcription. What message? That is which type of protein to be synthesized. That message taken by RNA from the DNA. So, that RNA that receives the message from the DNA, which type of protein to be synthesized, that RNA is called messenger RNA, because it carry message from DNA to protein synthetic area. So, formation of mRNA from DNA by receiving the genetic information, which type of protein to be synthesized, that information called transcription. In case of the prokaryotes, both transcription and translation occur in cytoplasm. But in eukaryotes, transcription occurs in nucleus, translation occurs in cytoplasm. So, anyhow, in transcription, mRNA is formed from DNA. DNA is the director, that is the master copy. RNA is working copy. Any type of RNA that is formed from the DNA only, either ribosomal RNA or transfer RNA or messenger RNA, formed from DNA only, and that process is called transcription. So, formation of RNA from DNA, that is transcription. Generally, we are using this word transcription during protein synthesis for getting the information from the DNA by mRNA.